All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. I think you're going to love this study today. The study's summary is that vaccinated individuals have immunoglobulin A and immunoglobulin G in their nasal mucosa and in their oral cavity, and that is aerosolized. That means it is present in the aerosol that comes out, the tiny microscopic water vapors, and it transmits to the people in the surroundings. And they tested the children of the vaccinated individuals, and their children had IgG present in their noses. So that is a summary. Now, of course, when I was here and I was looking at the comments, there were some comments as, how long will this IgG stay? So our nasal mucosa, our oral mucosa, and the mucous fluids here, they replenish or they exchange and replace very fast. You can actually see that you swallow your saliva and it is exchanged. The whole layer of cells present in this area get replaced within 7 to 21 days. So the structures that are present here, the proteins and the fluids, that are changed very, very often. Now, in this study, the author's point of view is very interesting. Their point of view is that possibly vaccinated individuals are providing passive immunity to the people around them. I think it's kind of a stretch, because a few breaths that have the immunoglobulins and they end up on another person's nose, in the nose and mouth. And these antibodies like virus, virus can replicate and increase in numbers. Antibodies cannot. So it's not really the passive immunity, but they conjectured that this may be passive immunity. So that was funny. Now, I also want to clarify, there is a concept of vaccine shedding, which I think in the more layman terms, we say that vaccine shedding is, in case of messenger RNA, that either messenger RNA or the spike proteins made by the vaccine are shedding. There is no study yet that shows the spike protein or the messenger RNA shedding from the aerosol. We have seen this study where the mRNA is present in the breast milk, but we haven't seen aerosolized mRNA or the spike protein itself. So this is not about vaccine shedding in those terms. But if we talk about antibody shedding, then that is what we're talking about. And honestly, if somebody is infected, their mucosa, the wet surfaces, will also have IgA and IgG and the virus as well. And their aerosol would also contain these antibodies and the virus. So it's nothing new. It's not novel, but it is interesting study. So I thought you would enjoy it as well. And I took a lot of time today to draw some pictures as well. My artwork is becoming better and better. So if you have any commissions, tell me. I think I'm becoming an amateur artist. Okay, let's start. So these are the gifts for humanity, and they're continuing very quickly. Let me show you the study itself. So this is drbeen.com. In the description of this video, there is a link where you can get the access to drbeen.com. Majority of these good lectures end up on drbeen.com. Plus, we have these awesome 3D anatomy atlases and a lot of beautiful lectures. You can go to plans, and if you just want to have access to the videos, that is the central one. It is a one-time payment. There is nothing further. But if you want to have CMEs as well, then it's $35 per month or $300 per year. So that is the drbeen.com. Now here is the study, evidence for aerosol transfer of SARS-CoV-2 specific humoral immunity. Beautiful study. I love it. And it is printed in ImmunoHorizons. Then some supporting links here. This is antibodies and their receptors, different potential roles in mucosal defense. Why do I have this link open? The question I hope will be in your mind, and that is, Where do these antibodies come from? Are they coming from the blood and then crossing the cell layers of mucosa? Mucosa is the wet surfaces, and then ending up on the wet side. How do they reach there? Or are they actually produced on the surface of the cells? And if that even a possibility? Or are these produced in the bone marrow and then travel all the way to the wet surfaces? And what is that pathway? So because of that, I have this link in here as well where they discuss that. And I would discuss that as a extra detail near the end of this lecture. Then I have some more links over here, the structure of secretory and dimeric immunoglobulin. This would just support the work I'm doing. Then in this study, there is going to be a term used, BSA. BSA stands for bovine serum albumin. So they use that as a control system. We'll talk about that as well. And then here is another BSA discussion. So with this, let's go to my drawings and the discussion of the study. So as you can tell, this is a person who is exhaling, breathing out, and has aerosol. You don't have to blow to make these aerosols. They are formed every time we breathe. However, when we are coughing or sneezing or laughing, then the aerosols are more in volume and more in velocity as well, so they can go farther. Like a child that is going to receive that aerosol. So a couple of texts from the study and then my drawings. So here they're saying the vaccines against SARS-CoV-2 have maintained remarkable efficacy against severe disease and death in those vaccinated, regardless of variant emergence, Omicron included. Less appreciated than the systemic immunity. So systemic immunity is the immunity generated within the blood circulation system and interstitium or within the body. They're saying less appreciated than the systemic immunity generated by the vaccines are the high levels of antibodies IgG and IgA found within the nasal cavity and saliva of the vaccinees. You can actually replace infected as well, because infected would have the same mechanism. It's not a different mechanism. So interesting that they say IgA and IgG on the wet surfaces of the body aren't really looked at very carefully. Then they say respiratory transmission of viral infection is proof that oral nasal cavity constituents can be communicated through aerosols and or respiratory droplets. So they say that, of course, we know that SARS-CoV-2 can go from one person to the other, mostly through the aerosol. As such, it would stand to reason that antibody present within the oral nasal environment may also be aerosolized to some degree. And they're correct in this. And it is already known, but they're going to do that for COVID vaccinees as well. And I want to show you the results before I go into the beautiful diagrams that I have. So results are the extended mandates for mask wearing in both social and work environments provided a unique opportunity to evaluate the possibility of aerosolized antibodies expiration from vaccinated individuals, meaning coming out of in the breath of the vaccinated individuals and getting stuck, these antibodies, on the masks. Utilizing a flow cytometry-based MMI to detect SARS-CoV-2 specific. So they did something to find if there are antibodies, and I'll explain that in more detail. They said that we found on the masks the antibodies. Consistent with the results reported by others, we identified both IgG and IgA in saliva from vaccinated individuals. It was therefore not surprising to detect both IgG and IgA following elution from face masks as well. So this is the result. Now I want to explain how they did the study. So there are 3 stages of experimentation here in this study. In the first stage, the researchers wanted to prove that the antibodies generated because of vaccines will be present in the mucosa. So IgA and IgG. So some of us may be kind of wondering that IgA makes sense. We have IgA protecting our wet surfaces, but what about IgG? So IgA is present in almost all wet surfaces, that means nasal area, mouth area, the respiratory system, the GIT, the genitalia, the urinary system, the bile ducts, etc. They all have IgA on them. These are all wet surfaces. However, IgG is also present, and I'll show you how near the end of this talk. Interestingly, I would like you to remember this, please. In male and female genitalia, in the bile system, and in the respiratory system, IgG is a predominant antibody on the surface of these systems than IgA. So first, they had to prove this, and I'll show you how did they prove it. Then once they had proved that, all right, on the surface of the wet surfaces, meaning if you have your saliva or nasal secretions, there is IgA and IgM. The second thing was that these are getting aerosolized and getting out. So the second proof was that, are these becoming airworthy? Are they getting out? So what they did was they collected the masks from healthcare workers who were wearing the masks and working and were vaccinated. And they collected the antibodies stuck on the masks, and they were able to show them. So that was their second proof that, yes, these antibodies are flying out of the oral and nasal cavities, of course, through aerosolized, and are now stuck on the mask. Then the third thing was to prove that the recipient, whoever is receiving the breath of this person, are these antibodies present in their system? So if they could prove these three, then they can prove with evidence that, number one, antibodies are formed and they are present on the wet surfaces. Number two, they are getting aerosolized. Number three, they are present on the people in the surrounding. So the third part is when they took the nasal swabs from the children of the parents who were vaccinated, and they found IgG in the children's nasal swab, and children were not vaccinated and neither were they infected. So that was the interesting one. So let's see, how did they prove these three things? Number one, prove that antibodies are present in plasma, saliva, and nasal mucosa. So what they did was they had volunteers. From those volunteers, they collected their saliva sample, their nasal secretions, and the blood plasma or blood. What they did was, to prove that there are antibodies in there, the researchers created little beads. And this is a very common standard thing. What you do is you take tiny beads, plastic beads, very microscopic spherical structures. On top of those beads, if I can make this a little bigger, on top of these beads, you can stick various kinds of things of interest, proteins of interest. So for example, in these beads, they stuck receptor binding domain of a spike protein. So if the blood or saliva or nasal secretion of the person has the antibodies against it, then those antibodies will come and get stuck on this area. They also stuck nucleocapsid protein. So if somebody was actually infected and they had the antibodies against the virus instead of the vaccine, then the nucleocapsid antibodies will come and get stuck on the nucleocapsid. They also had tetanus toxin present as a control as well, and they had bovine serum albumin as a negative control as well. So they had a multiple set of proteins against which they were testing for antibodies. And of course, we humans will not have bovine albumin in us. So if their testing experimentation was just flawed, then even they will start finding the bovine serum albumin. But if they would not find it, which they should not in humans, then they would know that we are doing the correct testing, and so on. So they created these beads, and on the beads they had receptor binding domain, they had nucleocapsid, they had tetanus toxin and bovine serum albumin. And now they created small wells. In my diagram, these beads are all sitting next to each other, but that's not how these are. Specialized beads are put in a different well and then exposed to saliva or nasal secretion or blood. And then they're going to test if the antibodies are present. So first set of the preparation. Then what they did was, on these beads, again in separate wells, they would put the fluids from the nasal secretions or from saliva or blood plasma separately. I just made them over here together just for the demonstration purposes or illustration purposes. And now if these substances have the antibodies against, for example, receptor binding domain, then those antibodies will get stuck to these proteins. So what they will do is then they will wash the beads, and the antibodies will stay stuck because antibodies are kind of now binding with the protein that is already present in the bead. So the rest of the junk will be washed away while the antibodies, microscopic, will be stuck to the beads. So imagine like this. Now the next thing is, how do they find out if these antibodies are present? So the way they do is, and this is again a very standard process, what they do is they have antibodies against the antibodies. So when they put those antibodies against the human antibodies, then they can detect if this well or plasma, or sorry, this little structure or substance has the antibodies or not. So they were able to prove with statistical significance that the saliva and the nasal secretion of the people who were vaccinated had IgG and IgA present in them. Of course, they also demonstrated the presence of IgG and A in the blood as well. So these IgG and A's could come from the blood. They could be present formed in the local lymph nodes, and from there they can come in. But they were able to prove the presence of these antibodies in all these three samples. Good. So point number one is proved, that a person who is vaccinated has antibodies on their wet surfaces. Now, the next thing was, the question is, will they fly? Will they get out? So for that, what they did was they took masks from healthcare workers. And what they did was they punched those masks, they broke those masks, with the punching machine, the manual punching machine that we use to make holes in papers. They used those punching machines to make 6mm holes really in the mask, but really what they wanted to have was those tiny pieces of the mask. They wanted to shred the mask into smaller pieces. Then what they did was those small pieces were put in another substance called elution buffer. And where these the substances that are stuck to the mask, they will become adsorbed in the fluids in this buffer. So now the mask pieces are present in this solution, and now they are adsorbed there. Then they will take that solution and also put those solutions on these beads. So now if the mask had antibodies stuck on it, then those antibodies will get stuck to the receptor binding domain or nucleocapsid or whatever other proteins. Very interesting, right? Very clever. So they were then able to prove, when they washed the beads and they looked at the antibodies, they were able to prove that the masks had antibodies stuck on them. That means these antibodies were aerosolized. They were flying off of the nasal cavity and the oral cavity, and now they were stuck even through the respiratory system, the bronchial airways, and now they got stuck on the way on the mask. Then the third thing to prove was that, will this now end up in the noses and mouths of the people who are in the vicinity of this person who is breathing it? So to prove that, they took the nasal swabs from the children of the vaccinees, and the children were known not to be vaccinated and were known not to be infected because they did not have nucleocapsid antibodies. So they made sure that they only included those children who were not infected and not vaccinated. And then their nasal swabs were treated the same way as I showed you the remaining things. And guess what? Children's nasal swab showed IgG present on them. So this is my diagram. Look at the, the diagrams I'm making nowadays. So children, this is the IgG hanging out of the nose, but really not hanging out. These are hanging in. So the nasal swabs of the children had IgG present in them. So this is the study. From this study, they were able to prove that vaccinated individuals will transmit IgA and IgG in the aerosol of their nasal and oral vapors, and that would go into the recipients and it would get stuck to the mucous membranes of the recipients. Now the question is, how does this happen? So this is a very important thing. So imagine these are the cells of our wet surfaces. So if you touch your tongue or your nose, the cells that we used to, remember we used to take the buccal swabs and there will be cells that would come off. So these are called the epithelial cells. So the red ones here are the epithelial cells. This little side here is the blood side. And this side where this blue colored mucus is present or fluid is present, that is the wet surface that we can touch. So what happens is, now this is very important. What happens is on the blood side or in the blood that is coming to these tissues like tongue and nasal areas and the airways of the bronchi and the GIT and bile and genitalias and urethras, urinary system. In this blood, there is IgA. So if you see here, this IgA is called monomeric. It is one molecule of IgA, immunoglobulin A. Monomeric immunoglobulins do not cross the cell barriers and end up on the saliva. They stay within the blood side. Then there are IgAs. There are two IgA molecules that are joined together with a protein called the joining protein or J protein, this red one over here. And then there is another component. So if you see here, this little blue handle, there is another protein called secretory component that is bound to them. What are we doing? Our body is preparing for this bivalent IgA, meaning two molecules of IgA, to be released to go to the wet surface. So first it joined them by a J chain. Now it is putting a handle on them, which is called secretory protein. The epithelial cells, the cells on the tongue, the nasal cavity, and all the other surfaces I said, epithelial cells, if you see here, they have specialized receptors that can hold or that can bind with the secretory component. This is how the cell would trap an IgA molecule, which is double molecule with a J chain and the secretory component. And then the cell will take that unit in, and the unit would pass through the cell like a boat going through the river, or a submarine going through the river or ocean. And then on the other side, it will come out and it will open up, releasing the antibody in the wet surface. What a beautiful mechanism, isn't it? So it's not that there used to be a thought that between the cells is a gap. And there are tight junctions here. There used to be a thought that the antibodies pass through this gap. That is not the case. Antibodies actually pass through the cells like they would cross an ocean bound in a submarine. And then on the other side, they would come out and they would be then in the fluid. Now that is about the IgA. How about the IgG? Now IgG has a special mechanism as well. It has special receptors that can identify IgG, and they would bind it and create a small pocket, which is called pinocytosis. The process is called pinocytosis. This small pocket is then going to cross the same ocean like the IgA did. The problem is that IgG is not supposed to be crossing this ocean. IgA is supposed to be, because it has a secretory component. You can think of secretory component as a visa to cross a cell. But IgG does not have that visa. So it is possible that within the cell, it will be attacked and digested and destroyed. So to prevent that, once the IgG is going through this cell, there are special receptors inside here that scan the protein and make sure that it is IgG. This is like, the X-rays on the ports, which look inside the containers to make sure it is the thing that is the correct thing to move. So the cell would scan the IgG molecule to make sure it is IgG. If it is IgG, the digestive enzymes are disabled from attacking the IgG, and this submarine also passes through the cell and goes and opens up on the other side and becomes part of the mucosa or the wet surface. Then, of course, as we breathe, the air will pick up a very tiny microscopic part of this fluid, and with all the contents of that fluid, which could contain viruses and bacteria and the IgA and IgG, and it will make a tiny little bubble from it, and that bubble will be expelled. This aerosol will be expelled, and you can actually see your breath expelling with those bubbles in wintry times. And now it is going to go into the other person's mouth and nose if there is a person nearby. And that is how these proteins end up in other people's mouths and noses. Can you imagine this? So very quickly, here, this is a diagram where, if you see here, this is IgM that doesn't go out. Sometimes IgM goes out as well if the secretory component gets attached to it. IgA monomer doesn't go out. Polymer or dimer becomes attached to the secretory component, passes through the cell and comes out on the other side. Then if you see here, IgG binds, pinocytosis occur. There are special receptors for that. Then it is prevented from destruction, and then it's going to come out on the other side. Then over here, it does whatever function it has to do. That is one. Then if somebody is interested in what is the structure of IgA secretory system, these are two IgAs that are combined together. Between them is a J chain, and then over here is the secretory component. And as you can see, it goes from the blood side to the wet surface side. So that is the discussion. So shedding happened, yes, but it is not the shedding of mRNA or the spike protein, at least not the studies that have shown it. Maybe there will be studies in the future that would show it, just like we have mRNA present in the breast milk. But this is not that study. This study shows, if we can call it shedding, normally we just say aerosolization. So this study shows aerosolization of antibodies from vaccinees to whoever is around them. That can be an unvaccinated person or a vaccinated person. So this is the discussion. I hope you liked it and you liked the drawings as well. Let me just very quickly scan for any questions here. And hello to everyone and happy weekend as well. So Doug says, this is exactly what I did. Is this spike shedding? No. So spike shedding will mean that the spike has to be formed and come out in the wet surface and then become wrapped in a bubble and then go out. This is not that. Although we talked about mRNA present in the milk. So there you could say mRNA. And if the mRNA is present in a cell, that cell might make spike as well, which would also be present in the milk. So that could be called shedding through the milk, but this is not spike shedding. Tech Etcher says, now with these submarines, I'm visualizing Asimov's Fantastic Voyage. John says, so OSHA was right. Unvaccinated wear a mask, check. Well, if they want to prevent the antibodies from coming in, then yeah. Thank you very much, Keeney, for your support. And if you would like to support as well, there are links in the description. You can become part of Patreon or Substack, or you can become part of this channel. We do on every Wednesday 2 Zoom talks only for the supporting beans. And we do 2 live Zoom sessions, one at 1 p.m. Pacific and the other one as at 6 p.m. Pacific. And we talk, for example, this time we discussed the myocarditis and troponin and exercise, and can myocarditis be mild or not? And so we discussed all those topics. So if you would like to have those more closer discussions with less censorship around us, then join me on Patreon or Substack. Let's see what else is there. Gary says, thank you. You're very welcome. So I'm Gregory. So see, within the same group of cool beans, there are varying concepts. So M says that that means maybe we should not wear a mask so that we can get the antibodies. Absolutely. That can also be the case. So McLevin says, could the vaccine be made only to make the S2 protein so it wouldn't be harmless? It would be harmless. Yes, you can. So it's really the RNA, which is a piece of genetic material. So you can make whatever you want. you can watch the replay. John says, don't wear the mask that have been punched holes. Absolutely. Rob says, fascinating process. Yes. Do you know that folks who have been here, so there are many other presenters. There are presenters like Matt Cram. He would talk about very selective things and lesser mechanisms. There are presenters like Campbell, John Campbell, and he has gone in a very different direction, more newsy, reporty, sensational. There are folks like Sunil Dhand, who is more also, the overall bigger picture of the processes and procedures and the ethics and morals. And there are then others too. I think we are the only group who for 3 years have now read immunology to such detail that we can have these discussions with doctors and immunologists and research workers and understand what they're talking about and maybe even know more. But most importantly, I think it gives us readiness for our own health and the health of our loved ones. And thank you very much for your support, for making this happen. Once again, links are in the description. Nice weekend. I'll see you next week. Bye for now.